Hello everyone, my name is Danny Slee and welcome to the RACI New South Wales webinar on forensic chemistry, fighting crime in the community. Just a few words to start. We have a late program change. We had 3 speakers, but we now have 4 speakers as of this week. So Marilyn Mckee is joining the team as well, and she's going to give a talk on application of NMR in the Australian Forensic Drug Laboratory. So now we have a total of 4 great speakers for today. The last webinar we had was in September 2020, and that was a microplastics in the environment webinar, and we have quite a few planned for the future as well. So we are planning for this year another webinar probably in June on a forensic subject as well. We also have some webinars on allergens and quality, and we are planning a face-to-face conference early next year, all things going well, and that will be called Healthy Waterways 2.0. Fair to say forensics is a bit of an unusual topic for our group, but we're keen to cover the subject. We have a couple of forensic chemists now within our group, so we decided why not, we'll give it a go. Now, first, before I start with the speakers, I just want to give a little plug. RACI Analytical Environment Chemistry Group, we're a collection of scientists from academia, from government and industry, and we have students all with a mutual interest in analytical chemistry in the environment. We have monthly meetings, and our group regularly organises seminars like this and symposia. We encourage students with a honours and masters event that's held once a year, and please feel free to join. First, just go to the RACI website, become a member, and then you can look us up. We're the Analytical Environment Group for New South Wales, and come and join. We not only promote environmental chemistry subjects, we also have social networking amongst the group, interactions. It's a really good group. Now, again, before I start, housekeeping. We have muted all registrants, but we have a Q&A function. You can just see, just at the bottom, there's a Q&A function on most PCs. Press that button and you can ask any question while the talks are being conducted. We're not going to answer these until right at the end. So after all the speakers, there'll be a Q&A session, and I will be selecting some questions out for the panelists to answer. There is no gap between talks. We're just going to go one talk after another. The order is Jason Tran first, Marilyn Mckee second, Professor Shanlin Fu third, and Joshua Van Vorst fourth. Our first speaker is Jason Tran. He's a senior scientist from FAS, the Forensic and Analytical Scientific Services, part of New South Wales Health. And his title of his talk is Forensic Toxicology, Protecting Our Community. Jason will be talking about activities in his laboratory, and you'll also hear about NPS. You'll also hear this from other speakers. These are called novel psychoactive substances, and some may know as the older term designer drugs. So let's everyone welcome Jason Tran. So Peter, just switch over to Jason and we're ready to go.
Thank you, Danny. All right. Good afternoon, everyone. My name is Jason Tran, as Danny said. I'm a senior scientist in the Forensic Toxicology Laboratory here at FAS, which is the Forensic and Analytical Science Service. Just to start off with, I'd like to do an acknowledgement of country, as we always do in New South Wales Health Pathology. So a little bit about myself. I've been employed here now for the last 16 years. I was first employed as a technical officer in 2006, and then a scientific officer in the Drugs and Driving Toxicology Laboratory. In 2019, I took on my current role as a senior scientist in the Forensic Toxicology Lab. I'm here today to give you a look at what FAS, and in particular forensic toxicology, does in the world of forensic science. And with the help of a couple of case studies, how forensic toxicology is used to protect our community. So let's start with what forensic science is. As you can see here, this is how TV portrays forensic science. And I'm 100% certain that if not all, but most of you would have watched some of these TV shows. So I just want to clear a few things up first. Most of us who are not cops, we're just civilians, we don't have guns. That's the first thing. Secondly, forensics is very, very specialized. So yes, there is a part of forensics that will go out to crime scenes and others that will cut up dead bodies. There are part of forensics that will analyze for DNA or fingerprints. Some of us work in labs with glassware and test tubes and beakers and microscopes. I've yet to see a Bunsen burner though in all these years, but none of us do it all. We don't all go to the scene, collect the evidence, and then take it back to our labs to analyze ourselves. We then don't go down and knock someone's front door down and arrest them. The other thing is that we can't do things in an instant or by magic. We can't give you an answer in 30 seconds. It's just not the way it works. Nor can we tell you what that little tiny chemical or smell in the air is, where exactly it was manufactured and from what company. That doesn't work. It doesn't work that way. However, when I did pick my science degree in chemistry and forensic science at UTS, I actually did think it was going to go that way. And because I did think that, I think a few other people also thought the same. And, you know, some of us might have seen this going around the internet about what a forensic scientist does. And anybody in forensics or in a lab really will understand that bottom right-hand picture of what we actually do. So, what is forensic science? It's the application of the different sciences in legal proceedings involving criminal and civil law. Toxicology is the science or study of poisons, more specifically the study of adverse effects of chemical, physical, and biological agents on living organisms and the ecosystem. This also includes the prevention of such effects. Toxicology is one of the sciences, and I'm more specialized in analytical chemistry. At FAS, or the Forensic Analytical Science Service, there are 10 other laboratories here. So apart from us, there's the Illicit Drugs Analysis Unit, Chemical Criminalistics, and so forth. We also have 3 forensic medicine sites across New South Wales, being Sydney, Wollongong, and Newcastle. And that's where the mortuaries are kept too. So our main task in forensic toxicology is to work closely with the coroner and forensic medicine to help explain why a person has died, to give peace of mind to their family and friends. Now, this is written into the legislation, specifically the Coroner's Act of 2009. This determines which cases are reported to the coroner and decisions on who may or may not have an autopsy done. At autopsy, biological samples are taken from the deceased, usually blood and urine samples, which then get sent to our lab for analysis and report. We also get vitreous humor, which is the liquid from the eyeball, and occasionally get bile, liver, stomach contents, and on the rare occasion, some human flesh. And if you're wondering, decomposed human flesh or muscle tissue looks a little bit like beef jerky. Liver is also an interesting sample type as well, because when we homogenize it before we test it, it really turns people off pate. We also get non-biological exhibits, such as liquids, containers, vomit, even clothes and blankets sometimes. For example, if someone is found next to an empty glass or a suspicious liquid, they might get sent to the laboratory for analysis as well. So, let me take you on a bit of a time travel into what forensic toxicology laboratory has looked like over the years. This is the 1960s. We used to use a lot of solvents. There were a lot of color tests, and most of the testing done was for heavy metals and poisons. It was primarily testing on stomach contents, liver, and actual organs, looking for very obvious cases of poisons. Now, as we travel to the 80s, we can see that, there we go, the 80s, we can see that there's a lot less solvent being used here. More instrumentation was used, and we had the introduction of chromatography. Here, we started to look more into the detection of specific types of drugs and not just the class of drugs. And now we have today. Science and technology has developed so much in the last 30 to 40 years We have QTOFs now that can do exact mass measurement. We can look at structural components of drugs, and there is a lot less hands-on work, a lot less solvent is used, and it is much more instrumental and quicker. In forensic tox, we receive about 3,500 cases a year. Of those cases, the majority of them are what we classify as obscure, which means unknown. It might be a natural death, or perhaps someone has fallen over and there's no explanation as to why they've died. Traumatic refers to a physical form of death, which commonly relates to suicide or murder. Overdose, which is very important for us, because toxicology is the only way that we can help prove overdose, and when we do our toxicology testing. Then we've got motor vehicle accidents, drowning, and the rest of it. Our workflow at the moment, so concentrating mostly on blood samples, which is our main sample type. Samples will come into the lab. We will subject them to our comprehensive screening, which includes alcohol testing by GC and ELISA screening for different classes of drugs. And we've got 2 LCMS QTOF screens, one being our new rapid tox QTOF screen. Once screening is complete, we have a range of analytical methods that we can then send for confirmation and quantification. This might involve liquid-liquid or solid phase extraction before we analyze them using our LC triple quads, GCMS coupled with MPD, and GCFID. We also have a number of targeted tests that we can complete when required in specific case types or when routine screening fails to identify any compounds of interest. So, what are the challenges in forensic toxicology? There are many challenges in forensic toxicology, and the main issue is post-mortem blood, which isn't great to work with. The human body starts to decompose as soon as the heart stops. Once decomposition starts, it can have a great impact on matrix effects. Now, depending on how the person has died, if the person has bled out or hasn't been found in a while and is very decomposed, there can be very limited sample available for collection. Post-mortem changes can also affect the stability of drugs, and the concentrations identified can be difficult to interpret. Then we have drug toxicity, and how some drugs can vary so much between each other between the classes. For example, paracetamol is toxic at high doses, and we can find them in hundreds of milligrams per liter sometimes. Some fentanyl analogs, however, can be fatal in the picogram range. This requires us to develop screening and also quantitative methods that will suit a variety of drugs at different levels. We're expected to be able to analyze for trace levels and also very high levels, and this can be very problematic at times. The other, and some would say the most challenging and important right now in this modern day in toxicology, is to be able to identify the rare drugs, the new drugs, ones that aren't very common, and the ones that are just, being developed all the time on the black market, and to be able to keep up with them. They're called, as Danny mentioned before, novel psychoactive substances, or NPS, and that's something that Professor Shan Lu will be presenting about later. Okay, so our first case study. It's not really a case study per se, but more something we've noticed growing over the last few years. A few years ago, the laboratory was very common to see alprazolam, sold under the name Xanax. It's a benzodiazepine and acts as an antidepressant. It is prescribed to patients with anxiety disorders, panic attacks, and other such behaviors. It is typically a low-dose drug. Patients are given anything from 0.25 to 3 or 6 milligrams at a time, at no more than 10 milligrams per day. It can be highly addictive. It makes the user drowsy and lightheaded, but it can also cause hallucinations and therefore be abused as well. It is commonly used to mix with other drugs to help counter some of these adverse side effects, such as nausea or vomiting. In our laboratory, we are able to quantify alprazolam using our LCMS triple quad. Etizolam, something that is very similar to alprazolam, is a thianodiazepine derivative, and therefore a benzodiazepine analog. It's something that the laboratory has not seen a few years ago, but lately, over the last few years, we've seen a lot of. So you could classify it at that time as an NPS, even though it was invented quite a few years ago, it wasn't really used as an NPS until more recently. It's also been used as a substitute for alprazolam on the black market, as the black market is able to provide it quite easily. We have seen an increase of people addicted to alprazolam purchasing it from the black market. This has led to an increase in people overdosing on alprazolam, but not for the reasons you think. What has happened is that we have seen a dramatic rise in the detection of etizolam in cases over the years, which has led to investigations into the black market of alprazolam. People are purchasing what they think is alprazolam only to get etizolam, which isn't as potent as alprazolam, and therefore the users are not getting the same effects. They're thinking they're just building up a tolerance for it. In actual fact, there is very little alprazolam in the tablets at all. So they up their dosage, and when they get a pack of real alprazolam tablets, they continue that dosage that they were currently on, and they end up overdosing on alprazolam. So working with the appropriate authorities, we've been able to cast a spotlight on this fake alprazolam trade, and not only help explain why some of these people have died, but also potentially save many more by alerting them to the fake alprazolam that's on the market. The next case study involves a group of users, a group of cocaine users. It started with a group who purchased some cocaine, again, off the dark web. Cocaine is a highly abused illicit stimulant. It causes euphoria, an increase in energy and sex drive. It raises your heart rate and body temperature and can cause insomnia. Now, after using some of this cocaine, the users immediately didn't feel like it was the same as the previous times they had abused this drug. One of the users, however, was smart enough to alert for medical assistance. The group were presented to hospital with varying degrees of sedation, hallucination, delirium, and some were combative. Most were given some form of further sedation and anticonvulsants. The hospitals could not figure out what was wrong with these guys, and all their tests so far could not explain what was happening. So they got in touch with the Ministry of Health, and the Prescription, Recreational and Illicit Substance Evaluation Team, or PRIZE, was notified. Now, we work very closely with PRIZE as one of our stakeholders and partners, and not just the coroners as well. So we work with these guys. They took on the case and forwarded us the samples. What did we find? LSD. Some of the powder was also sent to FAS, and the Illicit Drugs Analysis Unit analyzed it to be a very pure form of LSD, which is very, very rare to find. Now remember, LSD is usually very, very low dose and spotted on a piece of paper. These guys had a line of almost pure LSD thinking it was cocaine. Now this information was passed on to the PRIZE team, who informed the hospitals, and appropriate care was then established for everyone there. Everyone then recovered, and we were able to then notify users out there about this so-called cocaine, which was LSD, and to look out for it. So I hope I've been able to give you an insight into how forensic toxicology helps to protect our community. Not only do we help grieving families have peace of mind and explaining how their loved ones have passed, but also in conjunction with other stakeholders and partners, we can assist in treatment and prevention to ensure some of these people don't end up dead before we can help. Over the last couple of years, forensic toxicology and the PRIZE team have also won a couple of New South Wales Health and New South Wales Health Pathology Awards for protecting our community. Thank you, everyone.
Okay, hello. Thank you, Jason. Really interesting talk, actually. Quite fascinating case studies there. Now, without further ado, our next speaker is Marilyn Mckee. Like I said, forensic drug analyst for the NMI AFDL, which is the Australian Forensic Drug Laboratory. So she will start very soon. I'm just switching over now. Yep.
Is your sound off? Yep, cool. All right, let's share the screen. Cool. Okay, hello everyone. My name is Marilyn and I'm a forensic analyst at the Australian Forensic Drug Laboratory. And today I'm going to share with you how we help law enforcement fight crime with NMR. And thank you, Jason, for your presentation, because it's really cool to see the kind of similarities that we have in our work. So who are we? Let's see if I can change the slide. Yes, I can. We are the Australian Forensic Drug Laboratory, and we are a division of the National Measurement Institute, and we provide a range of services to law enforcement and government agencies. Now, in contrast to Jason, we receive materials that are just seized materials. So we do not have materials that have come from any sort of biological sample. We are receiving the samples as they are seized by the Australian Federal Police, the Australian Border Force, and the New South Wales Police Force. So we analyse these samples with a range of techniques, and from this, we get identification, purity. Ooh, hang on, I need to move this down. Oh, there we go. Purity, technical comparisons, and we can determine the synthetic route or geographic origin of the drug. So we're looking at what the drug is, how pure it is, and where it comes from in terms of synthetic or grown or that kind of backstory for it. And we use a whole range of instrumental techniques to do this, but I am focusing on my personal favorite one, nuclear magnetic spectroscopy. This is a really important technique because it can give us both quantitative and qualitative information about the drug. Here, identification and purity quantification. And this is so important for law enforcement agencies to be able to know the story behind it. So firstly, to fight crime, you need to know what you're dealing with, aka what sort of drug are we looking at? So accurately knowing the identity of the drug helps the law enforcement agencies track what is happening and respond appropriately with legislation and where to put resources. There have been many trends of novel psychoactive substances, and in some cases, these have been overlooked by authorities due to not knowing what the structure is, the lack of information, because sometimes things just simply evade detection because they're new. One such trend includes the synthetic cannabinoids. Now, these aren't new. Early synthetic cannabinoids were made for medicinal purposes, and thus they're well documented, they're in the literature. But as they started to get more and more popular, there were so many new structures coming out. And there's not the documentation for that. So we needed a combination of high-resolution mass spectrometry and NMR to identify the unknown substance, especially as there's no reference material to compare it. You can't just stick it on an instrument and hope for a library match. And this was a problem that was faced around the world. And indeed, one such drug, JWH18, was sold on the internet as herbal smoking mixtures and was not under any restrictions, as one, they didn't think it was psychoactive in any way. You know, it's just a herbal smoking mixture. And two, when you analysed it, there's no illegal substance detected by GCMS or IR, so just couldn't find it. It was a lab in Germany that used a combination of high-resolution mass spectrometry and NMR to identify the compound, which was the start of the synthetic cannabinoid trend. While not as drastic as the synthetic cannabinoid group, getting the structure right, has been important for our lab in providing accurate information to the required agencies. So here we have a recent example of a U10 analog. We had this sample submitted to the lab, suspected to be U10. And the match, when you looked at the mass spectrum of it, did look like U10, but when we looked at the NMR, it showed a slightly different story. Here we have the two mass spectrums. So mass spectra, we have the one in our lab at the top, and we have the one from a reference sample from the Sweet Drug Library. And as you can see, they are very, very similar. But when we look at the NMR, and here is the NMR here, this is the U10, and this is the analog that we actually have. I'm going to zoom in here to the aromatic region as this one peak that was like, oh, hang on, this is not consistent with this structure here. You wouldn't get this singlet for this structure because of all the splitting here. But this proton, based on where it's positioned, achieves a broad singlet. And so we're like, hang on, we need to confirm this structure. And indeed, when confirmed by other 2D experiments, it shows no, this NMR, this same sample, is not U10, it's actually this U10 analog. And we're now seeing this U10 analog popping up in several different seizures with various other drug combinations. So it's important we get the structure right so we know, we can provide the information to the law enforcement agencies, what's happening, where's this coming from, kind of thing. Sometimes the identity of the structure might also reveal information as to how drug smugglers are operating. Law enforcement and government agencies, they need to know how people are trying to smuggle drugs in. One common way has been in the form of impregnation. So often you might find a drug is concealed in something else, maybe a liquid, We call that an impregnation. However, they're aware that we know that, and they are evolving new techniques to try and evade detection. And one of these techniques is to chemically mask the substance. So this brings us to the story of TBOC MDMA. We received a while back in the lab a 80 liquid of hair product, and it was a viscous light red liquid that was labeled as hair product, but it had no match with field tests nor conventional lab tests. However, it was thought to be very, very suspicious, and it was further analysed. When you did look at the GC trace for it, there was, big unknown peak, and there was also a tiny MDMA blip and another blip that was a MDMA precursor. So this is a pretty interesting molecule to look at, sample to look at, what's in it kind of thing. And sure enough, with a combination of techniques that included NMR and mass spectrometry, this was identified to be TBOC MDMA. So this is an MDMA molecule where it's been chemically masked with this TBOC group, which is a common group in synthetic chemistry, a protecting group, to chemically mask this drug. And 2D NMR experiments were very important here because they can show the different connectivity between proton to carbon atoms direct, and also proton to carbon atoms nearby, a few bonds down, and also the couplings. So to really hone in what is this structure. This actually is not the only structure we've seen. There was quite a few others that came from it. There's our, such as this methamphetamine with a p-tosyl group. So we know you can mask drugs, but what if you mask the precursor? So sometimes people will send things, they'll ship a precursor, so when it gets to the destination, you can use that to make the drug. What if they mask the precursor? And indeed, that has also been seen with this MAPA, methyl 3-oxo-2-phenylbutanoate, which is very similar to P2P. It's a masked version. And once again, it's important to be able to identify the structure, and that can be done with NMR. Because if you're looking at a GC, you're not going to pick this up. Because there's no match to library. Sometimes you're looking for a library match and there isn't one. So it's important to be able to identify new compounds and stay on trends. And indeed, this one is still very widely trafficked in Europe. And the TBOC before, our group was the first one to identify, and it was observed internationally. And the data collected from AFDL was really useful in helping other people identify it around the world. Now, we're going to step back a bit from identification. That's a very important, and what's really classically what NMR is used for. But now we have purity quantification. So we can use NMR to determine the purity of drugs. And this is really important from law enforcement agencies because in evidential work, so when we're providing evidence for court, knowing the purity helps the law enforcement determine what type of charge the person gets, whether it's a commercial quantity, and that's especially important if there's a bulk quantity. And just it gives us information about the seizures as well. So what we would traditionally do is a GC-FID, which is also still used, and it requires the use of calibration standards. So you prep your standards, 1 to 5 or whatever, and you've got to prepare all that to be able to then prepare your samples and do the analysis. With NMR, it's very fast, it's very efficient. You do not need this calibration standard. What you're doing is more of a dilute and shoot method. You're really just putting in a deuterated solvent and with an internal standard that the concentration and purity of, and you're really just comparing that area under the peak for the unknown and the known. And we use automation processes to speed up this quantification as well. So in our lab, we use, we quant several things by NMR, such as methamphetamine, cocaine, GBL, and related substances like other amphetamine type substances as well. And when I mention automation, I am meaning the sample pro robot. Now, this is something that was custom built for us. We're the first one in Australia to have this, and it really speeds up so much time and efficiency in being able to use this robot. Because what we do is we pre-weigh our sample, we put it here, the robot prepares, just adds the solvent with the internal standard in, and it transfers it to the tube with us. So that's really sped up and enables us to work so efficiently. And actually, through the use of NMR, so quantitative NMR, and this robot, we've been able to tighten our accuracy, we've been able to tighten our precision, and of course, efficiency as well. because, you know, there's the efficiency going from a chromatography method to, not efficiency, sorry, accuracy going from a chromatography method to an NMR method. And there's also further accuracy gained and precision gained from having a robot do it as opposed to manual preparation. Here is an example of how we use NMR to determine purity. We have, this is the internal standard peak. We use maleic acid for this. And here is our methyl group that we're using. So what we do is we're comparing protons to protons here. So we know the concentration of this, and we can use that and the area under the peak and the amount of protons under the peak to compare like to like the protons. And we can get a purity from that way very quickly, very efficiently. And also by doing this, we can, you know, take a quick look and double confirm the identity of what we think we've seen from the GCMS. So that's our beautiful second technique. The beauty of quantitative NMR is actually also the fact that you don't need the standard of the material quanting. So you can get a new drug we haven't seen before, you can put it on the NMR, you can use the same principles because you're using the principles of NMR rather than the specific standard to be able to quant it. And that's really good to not have to always have a structure, to always have a reference material, especially in identification as well, when we really just don't always have a standard, especially when things are new. So, in summary, NMR allows identification of unknowns, masked drugs, and analogs, and NMR improves efficiency and accuracy in routine purity quantification. And this enables AFDL to provide accurate and efficient services to law enforcement agencies to fight crime. Back to you, Danny. Thank you.
Thank you, Marilyn. As usual, a fascinating talk. And you've sold me on NMR now. I think it's great. Now, let's keep moving on. I'd just like to say, just a reminder, you can use the Q&A function to ask any questions of the panel members. And like I said, towards the end of after the end of the talks, we can select some questions and the panelists will be able to answer them. So please feel free to use that function, the Q&A function, Zoom function. Now, our next speaker is Professor Shanlin Fu, and he is going to talk about non-targeted analysis approaches for the detection of novel psychoactive substances, NPS, which we've already heard a little bit about. And Shanlin Fu is going to expand on this. Shanlin is from the Centre for Forensic Science, UTS Sydney. So away you go, Shanlin.
Thank you, Danny. First of all, is the right slide? Do you see the right slide? Is this the right slide, Danny?
No, that's not the right slide.
Pardon?
That's not the right slide.
Not the right slide. Let me try again. I don't know what happened. How about this one?
Yep, that's the one.
All right, thank you. Yeah, thank you, Danny, for the introduction, and I'd like to thank the organizer for this opportunity to present and share some of our work in the space of forensic chemistry. I'm from the Center for Forensic Science at UTS and teach and research in the field of forensic chemistry, forensic toxicology, and clinical toxicology. As introduced nicely by the first and the second speaker, Jason and Marilyn, and I think you all appreciate that it is very challenging to detect NPS, simply because, you know, firstly, the sheer number of it, more than 1,000 NPS is being reported to the annual UNODC early warning advisory system, and more NPS to emerge each year. So there's a lack of knowledge on the structure and metabolism for the new drugs, and a lack of database and the reference material as well to develop a comprehensive method for targeted analysis. But therefore, we need a non-targeted screening strategy to pick up those newer drugs. So I'd like to share with you the two approaches or techniques that we use. The first one is high-resolution mass spectrometry, and the second one is the old school technique, chemical color reactions. So these approaches have the capacity to tell us what drugs or drug classes that might be present in the sample. We can then perform confirmatory structure determination using those techniques as just described or presented by Marilyn, including NMR, high resolution MS, and so on. So the high resolution mass spec is really an essential tool for non-targeted analysis. It offers, it offers the ability to report accurate masses. And because of the mass defect phenomena, that will give a lot higher confidence to us to determine the structure. So the non-targeted HRMS strategy is really a two-step process. The first step is component detection. You have to find out if something is there. The second step is component identification. That's to determine what is the possible new drugs of use. For component detection, there are a number of approaches that we can use. For example, the differential analysis, and you compare unknowns to a blank or control sample if you have, and you try to find the abnormal peak, and that might be something that of our interest. We can perform product ion searching for those common product ions, neutral losses, and so on, and to detect if a drug analog of that class is possible, is present in the sample. Or we can use apply the mass defect filtering. So the mass defect is really the difference between the nominal mass, in this example is showing 276, and the accurate mass, which has that decimal point. So the mass defect is actually that difference, 0.1799, as shown in this slide. So, so the mass defect of compounds in the same class may have clustered mass defect despite possible larger mass difference. So this is the unique aspect of mass defect that we utilize. So it's a very useful data mining tool. For component identification, because it's a high-res mass spec, and it gives you a very accurate mass measurement. So therefore, you have a less, very reduced number of possible target hit if you're using the high-resolution mass spec. So when we study the fragmentation pathway of fragments, and again, because of the high-resolution mass spec phenomena, we can have more confidence on the elucidated structure of that fragment, and therefore the structure of the unknown. So there's also retain time prediction and some other tool that we can use, relying on the artificial neural network or machine learning. So I will give you a bit of a example on each technique that we use. So a couple of years ago, we published this review article in the analytical and the bioanalytical chemistry, which summarize the various approaches that I just mentioned, and also the top-down, bottom-up approach commonly used in non-targeted high-resolution mass spectrometry work. So this paper published in the DTA journal that relies on the mass fragmentation analysis. So we're dealing with phenethylamine, the 2C, the DOX, the N-bombs. These are all very potent and central nervous system stimulant and strong, very strong hallucinogen. So even a lot potent than the LSD. So we identified quite a number of common neutral losses, common radical losses, common product ions for those drug classes. And these common neutral losses, product ions, have the potential to detect drugs in the sample in the same drug class by using the bottom-up non-targeted screening approach. So we did the same thing for the synthetic opioids and published in this journal, Frontiers in Chemistry. So we studied several different classes of opioids in this study. The fentanyl, we have 14 analogs. The AH series opioid, we have 7 of them, U series and the W series and so on. So a number of diagnostic fragments common to the different opioid classes were identified. So when used in combination, these diagnostic fragments show potential for use as class predictor without relying on database or reference material. So this DTA paper is an example of the application of Kendrick mass defect analysis for non-targeted screening of synthetic opioids in equine urine, equine plasma. So the Kendrick mass defect is similar to the standard mass defect, but we have to focus on a subunit. In this case, example showing that is a methylene group, CH2. A lot of the analogs of NPS, they differ by varying length of the alkyl chain. So they have a different length of a CH2 unit. So the idea is that the structure that vary by these repeating units will have the same Kendrick mass defect, regardless of their nominal mass. So by using this kind of a technique, we again studied quite a number of opioid classes, as it are using those drug analysis as an example. We spiked them in equine plasma at a very low concentration, 0.1 nanogram per mil. And the Kendrick mass defect filtering was applied. The number of filtered candidates is less than 20, which is very manageable. So we can start to focus on those potential drug analyte. And then all of the spike analytes were detected by using this technique. The machine learning techniques were used to develop class prediction model and retention time prediction model in this DTI paper. So the naive Bayes architecture class prediction model and developed has a capacity to predict accurately 90% of the opioid classes that we study. And the Gaussian process regression retention time prediction model, again, has quite good accuracy, and more than 80% of the retention time was predicted correctly. And those prediction is within 0.1 minutes of the experimental retention time. So the next technique is, as I said, is an old school technique, chemical color test. Because this chemical reagent, they react with functional group of a compound, and therefore has the potential to detect drug classes, even if the exact chemical entities are unknown. So it offers a convenient and cost-effective screening tool for the detection of the constantly emerging NPS. Currently, there are not many effective color test methods available for NPS screening. So the first color test method we developed is for piperazine analogs. And if you have piperazines and you obtain this very bright orange red color, and so we analyzed, at that time, we have 6 of piperazine analogs. All of the 6 piperazines were picked up by this test. And we also analyzed 84 of other illicit drugs or therapeutic drugs and common cutting agent. There's no false positive. So sensitivity and the selectivity of the test are very good. So the second color test we developed is for cathinone. And in the presence of cathinone, and this reagent will produce a yellow color. The original published in a paper is require heating at 80 degrees for 10 minutes, which is not really good for in-field application. So we modified the method by introducing some kind of a catalyst, the halide salt, and the reaction will take place at room temperature, and it will complete within 5 minutes. So again, the specificity and the sensitivity are quite good. We tested 44 of the cathinone analogs, and the false negative rate is at 12%, and the false positive for those other non-cathinone drugs is less than 10%. N-bomb, and so it will give this, blue color, and the reaction take place at room temperature within 5 minutes. Again, we test, quite a lot of the N-bomb analogs, 16 of them at the time, and the false negative rate is very low, 12%, but the false positive rate for those non-N-bomb substances, less than 5%. We tested 154 of them. Fentanyls, and this is a test being developed last year, and in the presence of fentanyl analogs, the initial color change go from this light yellow to green color, but eventually turn into this blue color. In the presence of light source, UV light, and the reaction speed is increased, we can have the color change within 2 minutes for the initial color change. Again, the sensitivity and specificity of the test are very good. So these three methods, cathinone, N-bomb, and fentanyl, has been patented, and the cathinone test being commercialized in 2018 by a German drug testing manufacturer, ESA test. And so the other two tests, the commercialization, the licensing negotiation is kind of down the way. We try to commercialize those methods as well. So currently we are in the process of developing a multiplex, so a testing device that can detect multiple drugs of use of those NPS in a single test. First we have to translate the chemical reaction from test tube or well plates onto paper, and then we are hoping to develop something that can really save time and be more convenient for the people working in the field. So I'd like to thank those people who contributed the discovery or the findings of the work that I present today, and those researchers from UTS: Morgan, Daniel, Joshua, Laura, Melanie, Natasha, Ronald. And I'd like to thank my collaborator, Dr. Adam Colley from the Racing Lab, New South Wales Racing, and Dr. Mark Tatu from Australian Federal Police, Dr. Sergey Bitny from New South Wales, and Dr. Chris Forek from Agent Technology. So I'd also like to thank the Australian government for the funding support through the Global Connections Fund, the priming grants, the bridging grant, and to help us to commercialize, for our commercialization effort. Thank you.
Okay, thank you, Shanlin. Yep, that really expanded on the NPSs, and no, it was a really, really good and interesting talk. Okay, we have one last one. It's Joshua van Vorst. He is the technical forensic analyst from the Forensic Evidence and Technical Services Command, Crime Scene Services Branch, New South Wales Police Force. His talk is on the Technical Forensic Support Unit for the New South Wales Police Force. Bit of a different perspective with this one. A lot more field work and field measurements are going to be talked about with Joshua. And I had a little preview of his talk the other night, and all I can say is that he's working in sometimes what could be described as an uncontrolled environment. So
Yes.
let's go to Joshua right now.
Okay, thank you very much, Danny, and thanks for the opportunity to be part of this. It's yeah, it's a good opportunity. So yeah, I'm Joshua van Vorst, and I'm, like Danny said, a technical forensic analyst in the unit that provides support in areas of hazardous substances found at crime scenes, basically. So just a bit of my background is that I did a forensic science degree at UWS, doing an honours project where I examined pig tissue with methamphetamine injected into it to see how long it persisted in the environment, sort of beginning a project on buried bodies that may have drugs in the system to see how long they persisted in the environment there. Then I was a quality control chemist for 3 years before getting a job with the police as a crime scene examiner, collecting fingerprints and DNA. Then after 3.5 years of doing that, moved into the Clan Lab unit, which is what the unit I'm in now was, it used to be called, mainly going out to drug clan labs and doing field analysis and sampling at those scenes. And now as a technical forensic analyst, I'm one of the supervisors of that unit, and I've been here for 6 years. So this is just a breakdown of our unit. There's one team leader, 4 supervisors, of which I'm one of, and 8 crime scene officers. And all of our staff in our unit have tertiary level chemistry qualifications and have also completed forensic training to be able to collect forensic evidence as well from the scene. So we're a bit of a one-stop shop there when it comes to hazardous scenes. And we cover the whole state of New South Wales with that response, 24/7, running an on-call system with that. So a clandestine laboratory is just a place where illegal substances are manufactured, and they can range from drugs, explosives, biological or chemical weapons laboratories. And there's a large crossover between those different scenes, depending on what they're making. There's still a large crossover. So that's a lot of the time what we have to work out. What are they making there? Is it a terrorism related thing, or is it a drug related thing, or what? So by far the most commonly encountered scene is where they're manufacturing illicit drugs. And I suppose that's because that's where the money is. So that's just a couple of examples of some scenes that we come across. So some are sophisticated in a way, those fabricated vessels there, and some are just stuff everywhere where they're just trying their hand at anything, sometimes it seems, with limited success. So in New South Wales, we respond to scenes multi-agency. So our unit goes out as the chemists at the scene, State Crime Command, the Drug and Firearms Squad, they're detectives, so they do the investigation behind all those drug jobs, and Fire and Rescue New South Wales provide hazmat and the safety side of things at those scenes. And then if it did turn into homemade explosives or counter-terrorism incident, the bomb squad, terrorism command, and even the army may be involved as well. So our main role at a scene is, first of all, to attend and assess the scene for its safety, and then to render anything safe that might be an immediate risk. Then once that's all under control, any items of interest are described, tested, and sampled. And during that process as well, we're always having to think of what process is occurring, because that will determine where the investigation goes and which sections in the police need to be involved. And a lot of the time there's people in custody that we need to have a bit of an idea of what they were doing so that they can be charged appropriately and we're not doing the wrong thing there. We also are a bit of a clean-up crew with that as well, because of the hazardous chemicals at these scenes. We need to find out what they are for the investigation side, but also for the clean-up side so that we can safely remove the items from the scene and store them appropriately. And then in the back end side of things, moving on from establishing what we think they're doing, we provide expert evidence at court after receiving the results from the confirmatory lab at FAS. We put that together with what we've seen at the scene and determine whether they were or not making drugs. So that's the chemical side of things. And because we're also forensic trained, we need to record the scene as a crime scene as well. So video, photography, sketches and notes, and then collecting of forensic evidence, fingerprints and DNA, and any other physical evidence that may be there. And just on another note, I mentioned we've just recently changed our name to the Technical Forensic Support Unit. With that, we've increased our capability, and we're looking at getting our own GCMS on site here, just to assist with some screening of the samples that we collect, because some of the jobs are so large that we're collecting hundreds of samples from the scene. And as you can imagine, it takes quite a while at FAS for them to sift through all of that just based off our notes. So having been at the scene myself, I will be able to bring the samples back to the lab here, do a screen on the ones that I think are most pertinent to the case, and then give a condensed number of samples to FAS for confirmatory analysis. But that's still in the pipeline where we're getting that organised at the moment. And as I mentioned, we go to other hazardous substance incidences such as CBRN, post-blast and homemade explosives. The skills required all have chemistry qualifications. It's a physically demanding role, so we have to go through medicals to be able to do the job. The PPE is quite strenuous to wear for long periods of time. With the large jobs as well, you're on scene for a long time, many days even. Need to be able to use scientific instrumentation, and a bit of experience in that is helpful. Forensic aptitude, working long hours, and yeah. So where are clan labs located? Anywhere. We find them everywhere. And this is an example of an apartment where there were open liquids everywhere. So it can be very risky in cramped spaces like this where they've just balanced open jars and cups full of liquid in them, us not knowing what they are. Same here, more examples of apartments where they've been converted into laboratories. In houses, chock-a-block full of items. Even that red arrow there is pointing to inside the wardrobe of this house. The floor had a hole in it and down the stairs into like a basement, and that's where the laboratory had been set up. And on the right-hand side there, that shows the laboratory setup. Hotel rooms, rarely, but still has happened before. Just a quick, probably a personal use type scenario where they make a little bit of drug and use it themselves. Vehicles, these would be packed up normally. We haven't come across the Breaking Bad thing where they've set up a lab in the back of a camper van or anything, but Often they transport their equipment and chemicals in the back of hire trucks, even cars. So if they're driving like an idiot, the police pull them over and find all this stuff, so then we would get called to that sort of thing. Shipping containers are often converted into labs with ventilation drilled through the walls and things. Rural properties are common, and those ones are often the most large scale because they can get away undetected, basically, in a big shed, so they can do large scale operations there. And in garages, and I've put this one in just to show that some of them are quite neat and people take pride in their work sometimes, but most of the time it's like this, where there is just junk everywhere, and decontamination after that is often an issue as well. Industrial units, so anywhere basically in the backyard. This is an extraction job of drug from the cactus, and even in a public park we've had one. So the size varies greatly, can be up to 500 items, and the longest we've processed a job for is 8 days, and it's pretty taxing after 8 days of wearing a hazmat suit and breathing through a full face mask. You get pretty worn out, and the speed at which we do jobs is about 10 items an hour between describing, testing and sampling, and then all the segregation at the end for cleaning up the scene. Yeah, that's how long it would take normally. So the types of drug manufacture laboratories we see are synthesis ones, so where they use reactants or the precursors to make a product, and oftentimes they'll even go one step before that and use precursors to make precursors, because the precursors are often scheduled themselves on the legislation, so illegal to obtain. So if you go one step back, sometimes you can find chemicals which are readily available, but you can make your own precursors and then in turn turn that into drugs. Refinement is another type. In the Act, the Drug Misuse and Trafficking Act, manufacture in relation to a prohibited drug includes the process of extracting or refining a prohibited drug. So refinement by recrystallization or purification is often just to make the drug look nicer so that you'll sell more of it. So instead of having little crystals, you'll have long, big, nice crystals of ice. Refinement by extraction is, like it says, extracting the drug from a matrix. So bark, the cactuses, but not always natural. Over on the right-hand side there's a box of t-shirts which were being imported, and they were just extracting methamphetamine from the t-shirts by using a washing machine and collecting the runoff water from that. So the most common drugs we come across are amphetamine type stimulants, that's for sure. Methamphetamine's our front runner, MDMA and MDA are not far behind. So the biggest hazards that we come across are just that we don't know what there is there, and so we need to have our level of PPE and safety high at the start until we know what we're dealing with. So hazmat help us with that, and we determine the working zones, hot, warm and cold zones at the scene, depending on where the work is going to be happening and where the clean areas are. PPE starts most basically with our overalls, which are fire resistant, and APR. Our boots and gloves are also PPE. Then we ramp up to what we normally wear at most jobs, with the TIChem suits for splash risk. Then self-contained breathing apparatus is the next level up from that, and then fully encapsulated chemical suit with self-contained breathing for the ones where we really don't know what's going on. And the reason we don't just work in the fully encapsulated all the time is that it's too hard work, and you can only spend 15 minutes in a fully encapsulated suit at a time. So a large job would take forever. So it's a case-by-case basis based on what we see there. So the different categories are active labs, where there's a reaction taking place at the time when we turn up. So that needs to be rendered safe. Inactive is the next category, which is it's set up, but it's not cooking at the very moment you arrive. Stored and boxed, like those ones in the vehicles, and a historical lab, which we often go to and take swabs from various areas. And from the picture there, we'd probably take some swatches from the carpet to see what had previously been happening. So we do presumptive chemical testing at the scene with a variety of techniques. So our biggest workhorses are the infrared and Raman spectroscopy instruments that we use. And we also do some gas detection. That's a lot for safety, but the multi-ray we've optimized for cyanide, phosphine, ammonia, and oxygen, so that if there's gas bottles there, we can see what's in them. And those particular gases are important in different drug manufacture methods. We have some ion scan, but that's back at the lab here, so we rarely use them for a clan lab, but we've got it on in our arsenal. And we're getting a handheld mass spec soon. We don't have that one yet, but that will bolster our capability. And we're looking into maybe these portable GCMSs, but we're doing research on them at the moment. And then we also do some old school wet chemical tests, silver nitrate, color, precipitate tests for different acids, pH, and this all helps a lot with segregation at the end of jobs. Sampling chemicals, we always need to take a representative sample. So the jar there on the right is a 3-phase liquid, so a equal amount of each phase in comparison to the actual item needs to be taken in the sample, so that you can see a representative of what was actually there. And we can take washes and swabs and scrapings of residues that are at scenes as well. And swabs as the premises are also quite helpful, because if there are high levels in a place where there's nothing set up, you can tell if something had been set up. And with the, I'll just touch on the forensic stuff quickly. We try and match the forensic evidence with the chemical evidence. So the best sort of thing for that is the PPE located at scenes. So a chemical sample might be taken from a glove. We cut the finger off and collect that for chemical testing, and then also take a DNA swab from inside the glove to link the processes that you might be able to detect from the chemical on the finger, and also link it to a person. And fingerprints, if there's glassware at a scene, they are great for getting fingerprints off. And we do all that in the hot zone too, because everything's contaminated. It can't come back to a fingerprint lab for analysis for fingerprints. And just lastly, we segregate. We need to make sure everything's packaged safely at the end of the job, and it goes off to our waste contractors for appropriate storage. And that is me. So thank you very much.
Thank you, Joshua. That was fascinating, actually. And I'm just amazed with those clan labs and how many different places you can have a clan lab. Obviously, you can do it anywhere.
Wherever you like.
Maybe in a motel room is beyond me, but anyway, that is really fascinating. I think now I'd just like to thank all the speakers. That was just a fantastic talk and a good summary of forensic chemistry, real practical stuff, and what's happening in the forensic chemistry market at the moment. Thanks everybody, and that's a wrap, I think. See you later.
Yep. Bye.